till now we have discussed three chromatographic techniques, that is ion exchange chromatography, gel filtration chromatography and affinity chromatography. All these three techniques, the mobile phase is liquid or a solvent. Today in this lecture we are going to discuss a technique which is where mobile phase is gas rather than a liquid. So, this particular technique is gas chromatography. So, we are going to discuss about in particular gas liquid chromatography. Now, gas chromatography is the technique used for separation of thermally stable and volatile organic and inorganic compounds. So, gas chromatography, like if a particular analyte has to separate or has to partition between a liquid phase and a mobile gaseous phase, then it has to be in vaporized state. And so, only those analytes which are thermally stable and volatile could be utilized or could be separated in here. Now, there could be two kinds of gas chromatography. One is gas liquid chromatography, which accomplishes the separation by partitioning the components of a chemical mixture between a mobile gas phase and a stationary liquid phase held on a solid support. Then there could be another chromatography, that is gas solid chromatography, which uses a solid adsorbent as the stationary phase. So, in this lecture we are going to focus on gas liquid chromatography technique. Now, if we compare gas chromatography with normal or other liquid chromatography techniques, which is in terms of like say either low pressure or high performance liquid chromatography techniques, then all the general principles and the performance parameter applies here also. So, here like say for example, retention volume or retention time is a characteristic of an analyte, and that will characterize its particular, it is a characteristic of a particular analyte. So, if you have to find, if you have to know about or identify a particular analyte, then if you know the retention time or retention or volume or elution volume, you can identify that particular component from a mixture. So, then other parameters like we have talked about resolution, capacity ratios and all those things will be applying in here also. So, it is a similar kind, but the difference is that here only volatile and thermally stable components of a mixture could be separated. Now, gas chromatographs or the gas chromatography equipment is little different from the normal chromatography equipment. The major components of a gas chromatograph are a constant flow of a carrier gas, that is the mobile phase as we have discussed, and you have to control the flow rates of that carrier gas through different valves and flow meters. There should be a simple system of introducing the sample into the gas flow and then into the column. So, there is a sample inlet point which is very important. Then after that a column has to be housed in an oven, that is because you are doing all the experiment or separation procedure at a very high temperature, and oven should be temperature programmed. So, your column in gas chromatograph is based in an oven which is temperature controlled. And then finally, when analyte comes out of the column, then a detector, amplifier and data recorder system needs to be there to analyze the chromatography of a particular analyte. So, these are the important components of a gas chromatograph, and we are going to discuss all different components in detail. Now, here in this diagram, if you can see on your screen, it is a schematic of a gas chromatograph, and it very nicely depicts what are the different components of the gas chromatograph. Now, if you see from starting here, this is the carrier gas supply in a particular tank or cylinder. This carrier gas is supplied from here, from this tank, and as it goes through the tubing, there are pressure regulators, pressure meters, and there are flow meters which can regulate the flow. Then there is a sample inlet in here which lets the sample in a vaporized form to, so it will be around somewhere here, vaporization will take place. So, there is a sample inlet from where sample will be injected. Now, this could be a direct injection, as we will see later on, or it could be a automatic sampler could also be used in here. Then there is a column here. This column is it's a very long column, and since it's a long column, it is coiled actually here, which is not seen. So, in this oven there is a coiled long column, but very thin, diameter is very less. They are very narrow bore columns, and then this column length is required for proper separations, and then this column is comes out here. The analyte from the column will pass through detector, and then certain electronics and microprocessor amplifiers, and finally data recorder, and you will see the peaks in here as you see in other chromatography techniques. So, the system or the principles are same as per their partitioning between the gas and the liquid phase. These analytes will elute at different times as per their retention times or their distribution coefficient here, and so the separation will take place. And like I said, and every analyte on a particular standard condition will have a characteristic retention time or elution volume, and through which you can identify the particular analyte. Even if for an unknown sample, you can make kind of a standard curve, or you can run standard compounds, and then you can plot it like we have seen in other chromatographic technique, and then unknown could be unknown sample retention time could be calculated from that. So, this is how you can, this is a very simple way to depict how a gas chromatograph will look like, or what are the different components of gas chromatograph. All right. So, let us start with the first thing. As we have seen in the gas chromatograph, it is a carrier gas supply, that is the most important at the beginning. So, you have to choose a mobile phase, and that is a gas phase. So, a constant supply of a carrier gas with proper controls is a very important necessary aspect of gas chromatograph. Normally helium, nitrogen, carbon dioxide or hydrogen can be used. These are the most commonly used carrier gas or mobile phase. Obviously, hydrogen has the disadvantage of explosion danger. There are suitable flow regulator valves and means of reproducing the flow rate. I think all of you understand that flow rate has to be reproduced to reproduce a chromatographic separation, or to maintain a particular retention time or elution volume. If flow rates change, the retention times and the elution volumes also change, and then you cannot compare the two experiments. A typical meter like soap bubble meter can be used for this particular purpose. So, carrier gas is one part where you have to choose particular carrier gas as a mobile phase. Now, as carrier gas goes on, the next thing is the sample injection. Now, like I showed you, sample injection, like sample has to be many times certain chemistry is involved to prepare the sample, and maybe derivatization or other things might be required to vaporize the sample, because not all the compounds could be vaporized. So, this is a limitation of gas chromatograph. So, sample injection, when we have to do, what it has to be done is that the sample has to be vaporized, so that it can partition between the liquid liquid phase, that is stationary, and mobile gas phase. So, there are two ways you can do it. One is direct injection. So, sample could be directly injected, and it is the standard mode suitable for 95% of packed columns. Like we will discuss what is packed columns. Then sample is injected by a hypodermic syringe through a self sealing silicon rubber septum onto a glass liner within a metal block, where it is vaporized and swept into the column. So, what is done? There is a heat block or metal block which is heated to a certain temperature, higher than oven temperature maybe, and the instantly as the sample is put on there, it vaporizes and it is swept onto the column. Now, the block is heated at a fixed temperature which is sufficiently high to convert liquid sample into a plug of vapor. So, what you are doing is you are injecting a liquid sample which could be vaporized, and it is done very fast. Samples can be measured with a calibrated loop, then introduced into the flowing gas stream by means of valve. For capillary columns, we will see what are capillary columns as we go along. Reduction in sample volume is necessary, because capillary columns capacity is very low, and this will be accomplished by an injector splitter, where 1 microliter sample can be injected, but only say 0.01 microliter enters the capillary. So, what is done is, as you are injecting the 1 microliter sample, there will be a injector splitter actually, which will split the sample, where only 0.01 microliter goes to the capillary column and rest of it is vented to the waste. Splitless injection is required for very small amounts of sample. The entire sample is injected onto the open tubular column through a modified heated flash vaporizer. So, there could be many ways you can do it. Now, this is a very typical representation of typical vaporizer, where you can inject the sample in here. As you can see on your sample on your screen, there is a syringe which injects the sample in here through a rubber septum, as I was talking to you, and this is there is a heat block here. If you can see, this is your heat block, and this is heated to a very high temperature. So, that liquid could be converted to vapor instantly, and then once it is done, this will be flushed into the column, where it can then be separated. So, this is a typical vaporizer, you can say, a flash vaporizer. Then you can have do injection by, or you can load your sample by automatic sampler also. So, sample vials are a glass throwaway type vapor tight septum caps. These vapor tight septum caps could be taken. Now, the procedure of sample injection, if you see, what is done is the sample flushes the syringe with a new sample to remove traces of the previous sample. That is very important, that you do not kind of get contamination from the previous run then in the sample injector. The pumps, then what is done is new sample will be pumped into the syringe to eliminate any bubbles, and then will be taken in a precisely measured amount of the sample will be taken. Then it will be injected into the gas chromatograph. So, you have to be very careful when you are utilizing automatic sampler, and the automatic samplers provide highly reproducible results, and they are much more precise. So, you can very nicely load your sample through automatic samplers. Alright. So, this was about how sample injection could be done on the gas chromatographic column. Now, let us come on to the next part, that is gas chromatograph columns actually. The two basic types of columns which are generally used in gas chromatography, these are called packed columns, as we mentioned earlier. Another one will be open tubular or called capillary columns. Now, let us discuss each of them one by one. Now, packed columns, they are constructed from tubing of stainless steel or nickel or maybe glass. Now, remember, all of these things has to be heat resistant, because they will be kept in an oven at high temperature. So these are they can withstand that heat. The inner diameter of these columns range somewhere around 2 to 4 millimeter, and they could in length they could go, like I said, they are very long, up to 1 to 3 meters length could be there. Now, these columns are packed with stationary face which is coated onto an inert silica support. Now, they are packed means they are completely packed, they are fully packed, and the support should not take part in the separation, like as we have discussed in other chromatographic techniques also. So, it should be inert, it must be thoroughly inactivated when polar compounds are to be analyzed. So, liquid phase or stationary phase is on support, and support has to be inert, as we have discussed earlier also. Now, there are some commonly used stationary phase which are could be polyethylene glycols, methyl phenyl and methyl vinyl silicon gums, esters of adipics and succinic and phthalic acids likewise, which are available actually. There could be beta cyclodextrine based phases for chiral separations. Now, the most commonly used support material is salite or diatomaceous silica or earth we call it, which is treated so that hydroxyl group are modified to avoid support sample interaction, and this could be achieved by salinization of the support with compounds like hexamethyl disilagane. So, you can use this is most commonly used support. Columns are then dry packed under a slight positive gaseous pressure, and after packing must be conditioned for 24 to 40 hours 8 hours by heating to near upper temperature limit, where the carrier gas at normal flow rates is passed through the column. Now, during this conditioning the column should be disconnected from the detector, because you do not want to foul the detector to prevent and you do not want to contaminate the detector. Now, what happens is when you are conditioning these columns, there might be a situation where column bleeding is taking place, where you are losing stationary phase or support material due to heating, and in that case it will certainly contaminate the detector. So, that has to be done. So, before you a column is commercially provided or you make it for use, it has to be conditioned so that there is no bleeding or it is minimized. Now, special packing materials may be needed for a particular applications. For example, very tightly loaded glass beads are used for very rapid analysis well below the boiling point of the sample components. There might be sieved Teflon supports used where corrosive substances are handled. And then in gas solid chromatography, the packing material is an adsorbent like silica gel or a bonded phase support or a molecular sieve. So, you can have different types of support for different types of applications. So, packed columns are very commonly used columns, but then there are capillary columns, and now capillary columns are different from packed columns. They have an internal diameter of say 1 millimeter or less. So, they are very narrow bore as compared to packed columns. They are constructed of fused silica having higher degree of cross linking within the silicon oxygen matrix. There is a high tensile strength of silica tubing and permits the construction of thin walled flexible columns. For protection of thin wall against scratches, coating of polyamide is applied to the outer wall. Now, there are two types of capillary columns, and these are could be packed columns or open tubular columns. Now, when you say packed columns, these are having solid particles over the whole diameter of the column, like we have seen in packed columns, but open tubular columns they are different. In a sense, you have what you have is you have an open and unrestricted flow path through the middle of the column, divided into like for example, let me show you this on your screen. So, what you have is, So, when you say packed column, and if I have a very thin column or very small diameter column, when you say it is a packed column, then it is filled in, it is all over, like, so you have beads which are a small stationary support which is filling or which is all over, completely over the diameter. So, it is like filling the whole area in here and there is no open space in middle actually. But when you are saying open tubular column, then in open tubular column you will have beads on the walls, inner walls of the. So, you will have beads here, but and you will have beads here, but you have an open area in middle. So, this open area lets the gas flow without any problem. So, there is a clear difference between a packed column and a capillary column. So, hope you have able to understand this. All right. So, let us return to our discussion. With an open and unrestricted flow in open tubular columns, capillary columns, there is a there is a flow path which is quite unrestricted here, and the gas can flow very easily here. Now, there are different kinds of open tubular columns. One is called wall coated open tubular column. In short form, you can say WCOT, wall coated open tubular column. Now, here the stationary face is thinly coated directly onto the walls of the capillary. Then there could be another one called support coated open tubular column. Now, here in support coated, like SCOT, support matrix is bonded to the walls of the capillary column. So, here the stationary face is not directly linked to the wall of the column, rather it is linked through a supporting matrix. The stationary face is coated onto the support and support is coated onto the wall. Then there could be another one called porous layer open tubular columns, which is kind of support coated used for adsorption work. So, the two main are wall coated and support coated open tubular column. Now, in terms of capacity, certainly the amount of material which support coated columns can open tubular columns can carry will be higher than wall coated columns. So, commonly used stationary faces which include like we discussed earlier, like polyethylene glycol or methylene polysiloxanes and other materials are utilized. They are coated onto the supporting matrix to give 1 to 25% loading depending upon the analysis. So, the capacity of the support coated columns is very high, considerably high than the wall coated columns. There are certain advantages of capillary columns. The capillary columns with particularly open tubular columns, they are having shorter retention time, they have greater inertness, they have longer life, lower bleed which is higher in packed columns. They have higher efficiencies and greater reproducibility. So, these are certain advantages of capillary columns. Remember, but the capacity of these columns is very low. So, many times they could be only for analytical purpose rather than the preparative purposes. All right. So, we have discussed about the column. Now, column is contained in an oven which is temperature controlled. So, chromatographic columns, they are coiled and they are held in a basket that is mounted inside an oven. Now, column must be able to be rapidly heated and cooled in this oven. So, this requires a well designed and very adequate system of airflow. Ovens are usually constructed of low mass stainless steel and the working temperature range is chosen to give a balance between a peak retention time and resolution. Now, column temperatures is controlled to say plus minus 0.1 degree Celsius and the analyte partition coefficients are particularly sensitive to temperature. So, that is very important. Analysis times may be regulated by the adjustment of the column oven, which can be operated in different modes. Like say it could be operated in either isothermal analysis where a constant temperature is applied and should be maintained here, There could be temperature programming as well. So, you can have isothermal as well as you can have temperature programming, where the temperature is gradually increased to facilitate the separation of compounds of widely different polarity or molecular weight, where temperature should be maintained within plus minus 2 degrees Celsius. So, you can have both ways you can operate the oven, that is isothermally or by programming the temperature. Now, liquid phase, stationary phase or coated on inside the column or on the support material, as we have discussed, it is like here partitioning takes place, and it separates the sample components into discrete peaks. It should be chemically and thermally very stable, because otherwise, like we were talking about column bleed and other problems could occur, which will certainly damage or will result into not so good chromatographic separation on gas chromatography. Operations are always kept 10 to 15 degrees Celsius below the upper temperature limit of the phase, because again column bleeding will be much higher if you go at the upper limit. Then the amount of column bleed must be minimized, and it will certainly prolong the column life, prevent any fouling of the detector, and maintain baseline stability on the chromatogram. So, that is a very important part, that column bleed could be minimized to enhance the quality of the separation. Now, bonding the stationary partitioning liquid to the inner surface of capillary columns or to the column packing permanently anchors the stationary phase to the surface. Commonly used phases, which we have already discussed, includes polyethylene glycol, methyl phenyl and methyl vinyl silicon gums, esters of adipic, succinic and phthalic acids, epizone L and so on. So, there could be different kinds of depending on what temperatures, what analytes you are going to use, you can use different kinds of liquid phases. Once the analyte has passed through the column, it has partitioned and it has the mixture has different analytes in a mixture has separated. They will come out of the oven and they will be passing through the detector, like they will be eluted from the column and passing through the detector, so that they could be identified, or those chromatogram could be obtained. Now, detectors, there are many different types of detectors, as we will see. Now, detector is located at the exit of the separation column, and there are what it does is it will sense the presence of an individual component as they leave the column, and there could be different ways methods to sense that particular analyte in a vaporized phase. Now, volume must be small to prevent the remixing of the components separated, so that should reach the detector fast. The electrical analog output of the detector is amplified and then sent to the strip chart recorder, or converted to a digital signal and sent to a computer system. Then computer system can process the data, store them and display the chromatogram with analytical results on your screen or a recorder. And there are several types, like I said, detectors. Let's see, let's discuss some of these detectors here. There are very simple type of detector called thermal conductivity detector, or TCD we call it. Now, thermal conductivity detector are one of the earliest detectors actually, which were developed for use with gas chromatography. Now, what it does is it utilizes a heated filament placed in the emerging gas stream. So, the amount of heat lost from the filament by the conduction to the detector walls will depend on the thermal conductivity of the gas phase. I will show you that in a little while. Now, within a cavity in the metal block, there extends a tightly coiled filament, like I said, this is heated filament. Filament is heated to a constant temperature, but less than a dull red condition by a regulated current supply. Now, heat loss from the filament to the metal block is constant when only carrier gas is flowing through the detector. So, when only like certain carrier gas and there is no analyte in it, then the heat loss will be a particular amount to the walls of the detector. Now, the thermal conductivities of say hydrogen and helium are roughly 6 to 10 times greater than those of the most organic compounds. So, what will happen? If there is an analyte or organic compound present, the presence of even small amounts of this material, organic material, will cause a relatively large decrease in thermal conductivity of the column effluent. So, what will happen, that the amount of heat lost or which is conducted to the walls of the detector will be less. So, the filament will retain more heat and its temperature will rise. So, that particular its electrical resistance will go up. So, the advantage of, and you can measure that, the advantage of thermal conductivity detector is its simplicity, its large linear dynamic range, which is around 10 to the power 5, its general response to both organic and inorganic species, it does not differentiate between them, and its a non destructive character, like it is not destroying the sample. So, you can collect also the sample after detection. Limitation, one limitation could be it is low, it has low sensitivity. So, that could be one limitation. Now, this is a very simple schematic of this thermal conductivity detector. As you can see, there is a filament here. This filament is heated. Now, when it is heated and there is a gas flow here, which is only carrier gas, the heat generated from here will go to the walls of the detector. Now, when there is an analyte organic compound mixed with the carrier gas, then the conduction of heat will be lower, and that could be measured actually, because resistance will go up here, and that is how you will get the signal. Like we said, it is non destructive. So, flow out here you can collect your sample after it has been detected. So, thermal conductivity detector is one type of detector, but it has a low sensitivity, as we were talking about. Now, second most commonly used detector is flame ionization detector, or we call it FID. Now, FID are the most what you call generally applicable, and it is the most widely used detector, like I said. A mixture of hydrogen and air is introduced onto the detector to give a flame. So, it is a flame ionization. So, flame is an important part of this detector. So, a flame forms one electrode. So, what is done is a mixture of hydrogen and air is introduced into detector to give a flame, and the jet of which forms one electrode. The other electrode will be a brass or platinum wire mounted near tip of the flame. So, this detector will have one electrode in form of flame, another in form of a wire. So, what happens is when sample components will emerge from a column, they will be ionized in the flame, and when they are ionized, they will result in increased signal being passed to the recorder, and that will be measured. Upper temperature limit here is 400 degrees Celsius, and minimum detection quantity is the order of like say 5 into 10 raised to power minus 12 gram per second. If you compare with TCD, it was 10 raised to power minus 8 actually, which is lower than this. So, this has much higher sensitivity. Here one thing has to be remembered, this flame ionization detector, they responds proportionately to the number of CH2 groups. So, if you have 2 CH2 groups, then signal will be a particular amount. Then if you have 4 CH2 groups, it will be double of that, likewise. So, it is like directly proportional to the number of CH2 groups, but there is a problem that no response from the fully oxidized carbons will be obtained. So, the response from carbons attached to say certain groups is like say hydroxyl groups and amine groups is lower, and oxidized carbons will not give any signal. So, that is a disadvantage. Now, it is advantageous to use FID, that is flame ionization detector, because the detector is unaffected by the flow rate. It is non combustible gases, and it is not affected by flow rate or non combustible gases or water. The properties allow FID high sensitivity and low noise. The unit is both, it is very reliable and very easy to use, but you require a flame here, flammable gas here, and also the sample is destroyed. So, unlike a thermal conductivity detector where it was not destructible, this is destroys the sample. If you see the very simple schematic here of flame ionization detector, then you can see here there is a flame here and there is this collector, you can say collector assembly with a wire. So, what happens is as the exit or material is put in this flame, it ionizes and then current is passed and the signal is recorded, and like I said, it is a destructible. There are like grounded jet, there are a lot of hydrogen air flame is here, hydrogen is supplied from here, and air is supplied for the flame. So, this is very useful and most widely used detector, and like I said, it has certain limitation that oxidized carbons cannot be sensed here. Then other groups like hydroxyl will be have lower detection limits, and it is directly proportional to the CH2 groups. So, it is very useful for say environmental samples and a lot of like say in pollution studies and a lot of other studies it is utilized. Then another version of flame ionization detector is thermionic emission detector or TED. This is also called nitrogen phosphorus detector, and it is same as FID, but it has you can say much cooler as compared to the FID, and it has a crystal of sodium salt fused onto the electrode system or a burner tip embedded in a ceramic tube containing a sodium salt or rubidium chloride tip. It has an excellent selectivity towards nitrogen and phosphorus. It has been modified for compounds containing nitrogen and phosphorus containing analytes. Upper limit range could go up to 300 degrees Celsius. Like I said, it is kind of cooler or flame is cooler than the flame ionization detector. Detection limit is very high, say up to 10 raised to the power minus 14 grams per second. It is widely used for organophosphorus, pesticide residue analysis and other things. So, here there is a simple schematic, it is like FID only, there is detect here, there is a flame which is a it is a cooler flame, like I said, there is a heater connections, there is a rubidium or cesium bead here, anode which is collector here, and as the analytes are ionized here, then mostly nitrogen and phosphorus compounds, then the signal could be or could be recorded in here. Another detector which is for a particular kind of compounds is called electron capture detector. Electron capture detector only responds to analytes that can capture electrons, as the name suggests, particularly halogen containing compounds. It is widely used in analysis of say polychlorinated compounds like pesticides, DDT, aldrin, dialdrin and all those things. It has a very high sensitivity of around 10 raised to the power minus 13 grams per second, and the upper temperature could go up to 300 degrees Celsius. Now, this detector works by means of a radioactive source present on surface of one electrode, which will emit high energy electrons, that is beta particles. If you could recall, in radioisotope technique we have discussed about that radioactive substance will emit certain particles or radiation. Now, these electrons bombard the carrier gas, as we have seen in gas ionization based detectors there, releasing an electron, and that gives the current across the electrodes when a suitable voltage is applied. So, if you could recall GM counters, it is something like that. Now, what happens is when electron capturing analyte emerges from, so when you are taking carrier gas, the different current is recorded, but when you have an electron capturing analyte in the carrier gas, what will happen is the ionized electrons will be captured by these analytes, and there will be a drop in the current, and this change could be recorded. So, the carrier gas, so here that is the method, and cobalt could be utilized as a radioactive material, or other things could be also utilized. The carrier gas most commonly used in conjunction with ACD is nitrogen or an argon plus 50, it is a nitrogen or an argon plus 5% methane mixture. So, that could be either nitrogen could be used, or argon mixed with methane, and that is 5% methane could be mixture could be utilized. So, it is a very simple schematic of electron capture detector. There is a radioactive beta emitter, and there is a gas here, and there are electrodes here. Now, radioactive material will emit beta particles, which will ionize the gas, and you will have a current as you apply the suitable voltage. So, when there is no analyte in the carrier gas, there will be a current, and when halogen containing or electron capturing analytes are there, there will be drop in current that could be measured. So, electron capture detector also are very useful for analytes which can capture electron. Now, there are other detectors also. These were what we have discussed were the most common types of detectors, but there are other detectors also, like say flame photometric detector, which exploits the fact that phosphorus and sulfur containing analytes emit light when they are burned in a FID type detector, and this light is detected and quantified, and a detection limit can go up to 1 picogram for phosphorus containing compound and 20 picogram for sulfur containing compound. Then there could be like photoionization detectors, which uses ultraviolet radiation from lamps with energies ranging from 9.5 to 11 electron volt to produce ionization of solute molecules. The ions are collected at a positively charged electrode, and the current is measured. Compounds whose ionization potential are lower than the lamp ionizing energy will give a response. There could be another like electrolytic conductivity detector, which operates on electrolytic conductivity principle. So, organic compounds eluting from the column, that is gas chromatography column, will be burnt in a say miniature furnace to form simple molecular species, and that readily ionizes and contributes to the conductivity of deionized water, and this change in electrolytic conductivity will be monitored. So, we have discussed these are different types of detectors which could be utilized, and they could be have applications for different kinds of works or different kinds of scientific techniques, like say it could be in environmental science or medical or clinical research, where gas chromatography could be utilized, or in basic research. Like I said, the thermal conductive detector is very simple, very useful detector, and the most widely used is flame ionization detector. There could be many applications of gas liquid chromatography. There could be qualitative or quantitative. Like for qualitative analysis, generally chromatographic data is presented as a graph of detector response against the retention time, like you can make a standard curve, or you can plot them. It is called a gas chromatogram. So, this provides a spectrum of peaks for a sample representing the analytes present in a sample eluting from the column at different times. Retention time can be used to identify analytes if the method conditions are constant by comparing the retention time of the sample as well as the standard. And I have shown you just when we have shown I have shown you the gas chromatograph, the schematic of the gas chromatograph. Now, these pattern of the peaks obtained will be constant for a sample under constant or standard conditions, and then it could be identified from a very complex mixture of analytes. Then you can also test the purity of a compound here by comparing the standard and sample. If additional peaks are obtained, then impurities are present, and otherwise the compound is pure. So, if there is any additional peak, compound is not pure. So, the purity of a purified compound could be tested in gas chromatography. In most modern applications, the gas chromatography is connected to a mass spectrometer or a similar kind of detector, that is capable of identifying the analytes represented by the peak. So, the samples could be passed to mass spectrometer, and where structural information through molecular mass information you can identify the samples, and many more things could be done on a mass spectrometer, where a lot of information about the analyte could be obtained. In quantitative analysis, like for example, area under a peak, like we have done for like you can do it for other chromatographic techniques, where quantitative analysis could be done by measuring the area under the peak. So, here also area under a peak is proportional to the amount of the analyte present in the chromatogram, and by calculating the area of the peak using the mathematical function of integration, the concentration of analyte in the original sample can be calculated. Concentration can be calculated using a calibrated calibration curve created by finding the response for a series of concentrations of analytes, or by determining the relative response factor of an analyte. So, this is like you can do quantitative analysis. Relative response factor is the expected ratio of an analyte to an internal standard or external standard, and is calculated by finding the response of a known amount of analyte and a constant amount of internal standard added to the sample at a constant concentration, with a distinct and it will have a distinct retention time, like you have a distinct retention time to the analyte. So, these are few methods for quantitative analysis. They are quite similar as for other chromatographic techniques. You can use internal or external standards, and by various methods you can do that. Gas chromatography is utilized for a lot of different applications, like elemental analysis in different fields of biotechnology. Like you can determine say elements like carbon, hydrogen, oxygen, sulfur and nitrogen. You can use it for analyzing mixture of drugs. Isolation and identification of drugs could be done. You can have isolation and identification of mixtures of components, for example, plant extracts, volatile oils, amino acids, and a lot of other could be tested and identified and even isolated. So, there are a whole lot of applications for gas chromatography. With lot of applications, there are limitations, as we were talking. Here samples which are analyzed are limited, because not all the compounds can be volatile and thermally stable, but many times it can be made volatile by derivatization, where a lot of chemistry is involved. Sample must be thermally stable to prevent degradation when heated, because if it is degraded, then it is no use to purify it or analyze it. The gas chromatography cannot be used to prepare samples for further analysis once separated, because you do not get whole lot of amount. So, there are certain limitations also. Gas chromatography nowadays has been replaced by high performance liquid chromatography for many applications, but still gas chromatography is used by many different streams of biotechnology, and like for example, clinical analysis, environmental science, a lot of testing for say drugs, pharmaceutical industry, in like food industry, a lot of analysis is done, particularly of say certain analytes or certain elements is done in here. With this lecture, we come to an end of chromatographic section. So, in chromatography we have discussed lot of different techniques. We have discussed basic concepts, we have discussed about liquid chromatography, where low pressure and high performance liquid chromatography we have discussed, and we have discussed these 4 techniques. So, this completes our chromatographic section. In the next lecture, we will start a new topic, and that is electrophoresis, which is another very important analytical method, which is routinely used in various laboratories for both research as well as for application purposes. Thank you.